Good evening, viewer. Welcome to our FTB English. Here is the most amazing news for our viewers. Yeah, two heads are better than one, as the saying goes, and sometimes two instruments ingeniously recombined can accomplish feats that neither could have done on its own. Such is the case with a hybrid microscope born at the Marine Biological Laboratory that for the first times allows scientists to simultaneously image the full 3D orientation and positions of an ensemble of molecules such as labeled proteins inside cells. The research is published this week in Proceedings of the National Academy of Science. The microscope combines polarized fluorescence technology, a valuable tool for measuring the orientations of molecules, with a dual view light slit microscope which excels at imaging along the depth axis of a sample. Yeah, this scope can have powerful applications. For example, proteins change their 3D orientations typically in response to their environment, which allows them to interact with other molecules to carry out their functions. Using this instrument, 3D protein orientations changes can be recorded, said first author Talon Chandler of CJ Biohub San Francisco, a former University of Chicago graduate student who conducted this research partly at MBL. There is a real biology that might be hidden to you from just a position change of a molecule alone, he said. Imaging the molecules in the spindle of a dividing cell, a long-standing challenge at MBL and elsewhere, is another example. With traditional microscopy, including polarized light, you can study the spindle quite nicely if it is in the plane perpendicular to the viewing directions. As soon as the plane is tilted, the readout becomes ambiguous, said co-author Rudolf Oldenburg, a senior scientist at MBL. This new instrument allows one to correct for tilt and still capture the 3D orientation and position of the spindle molecules. Behind the exclusive report, FTB English, Delhi, Bharat.